as we go, but in the interest of time, I'm going to go ahead and introduce Kay. So welcome everyone to the Environmental Engineering Graduate Seminar. This will be our last seminar of the semester, so thanks for coming to all the previous ones. Thanks for coming today. If you want continuing education credits for this, just email Dr. Eric Segrin and he can get you a certificate. So today we're honored to have Kay Millerick. She's an assistant professor in civil, environmental, and engineering and construction engineering at Texas Tech University. She received her Ph.D. in environmental engineering from the University of Illinois at Urbana-Champaign in 2014 and a Bachelor of Science from Florida State in 2007. She's an experimental environmental microbiologist whose research works at the interface of environmental pollutants, reactive surfaces, and native microbial communities. She mentors a diverse research group at Texas Tech, is actually involved in efforts to improve gender equity in academia, and is the proud mama of a busy toddler. All right, with that, I'll let you take it away, Kate.
All right. Well, Brian and Michigan Tech, thank you so much for letting me speak today. I think my last slide has a photo of the busy toddler for anybody who is curious. But that introduction pretty much summarizes my lab here at Tech. I tend to do a lot of work with environmental microbiology when there's both microbes, pollutants, and some type of surface involved. My surfaces can go anywhere from carbon materials to different forms of iron to one of my projects is actually looking at different well casings and how that influences microbial behavior. The one that I'm going to talk about today is about contaminated sediments and different sediment capping systems and how those materials influence microbial behavior and ultimately pollutant fate and water quality implications. But before I talk more about today's project, I do have to go for a shameless plug, which is this seminar that Michigan Tech is running seems very similar to the seminar that Texas Tech operates. Every 2 to 3 years, I'm in charge of organizing speakers for that seminar. This coming spring, I am the seminar organizer at Texas Tech. So if you're interested in giving a seminar talk at a different university, if you'd like to see different exposure, we typically have about 20 to 30 student and faculty members attend the environmental engineering seminar here. Certainly, I would love to schedule someone from somewhere different just because a lot of our seminar speakers over the years tend to be a little bit repetitive. So if you are interested in giving a seminar talk, please email me, and I think my email is at the end of this presentation. So with that, I will begin my talk today on sediment contamination and whether or not development of biofilms leading into an actual bioreactive sediment cap or sediment top would actually solve some of the chronic issues we see with long-term sediment contamination. The work that I'm about to show today was the work of my doctoral student, Giovanna Pagnosi. I did spend quite a bit of time shortening this presentation. Several of these slides were from her dissertation defense. I figured no one here really wants to sit through an hour-and-a-half long dissertation defense, so it is significantly shorter than her presentation. I just tried to touch on key findings. One note about this presentation is that Danny Reibel, her co-advisor, did a lot of the mathematical computational numbers that are presented in her manuscripts. I'm not going to spend a lot of time on the math balances and some of the computational side just because that was largely led by her co-advisor, and to be perfectly frank, I'm not very good at explaining mathematics. I am more interested in the microbial side, so this presentation will slant a little bit that way. So with that, I'm going to go ahead and turn off my video. Sometimes it's a little bit weird looking at slides and also being recorded at the same time, but please feel free to stop me if you have any questions or if I'm just talking too fast. Video is off, but hopefully everybody can still hear me. Oh no, it's not letting me progress. Hold on.
I think if you go to slide mode, maybe not show slides, but just where you see the slides on the side It'll let you go forward or something like that.
Is that what it is? Okay, let me play with this. I do apologize.
We've had other speakers have the same issue, so it's some kind of Zoom thing, or I don't know. Yeah, try that mode.
So let me try this. Oh, there we go. Apologize. All right, so my presentation outline today, I'm going to generally be talking about an overview of sediment capping and issues with sediment caps, which at least for me was a slightly different spin on what I typically study, just because I don't typically study the sediment cap environment. This particular project stemmed out of the fact that Giovanna, the doctoral student, was particularly interested in sediment caps. And I'll be summarizing just 3 of her many projects that she worked on during her dissertation. Side note to anybody on this call who is a doctoral student, one thing that, as it turned out with Giovanna's presentation, that I would not recommend to anybody is we did wind up submitting all 4 of Giovanna's publications over the course of a 6-month period. And my one bit of recommendation for any PhD student is try to stagger them a little bit more than 6 months, because that 6-month window does get pretty rough. And so what I'll be showing here is the summary of those 4 publications, 3 of which actually have gone out. The 4th one is still under review. So if you do happen to be one of my reviewers, please be kind. Oh, it's not letting me, I'm so confused. It let me advance before. Oh, here we go. I got to click. It's not letting me use the arrows. Perfect. All right. So what we're going to be talking about today are contaminated sediments, which is a chronic issue globally. Typically, when you're looking at contamination within sediment beds, you're looking at either organic contaminants or inorganic contaminants that generally don't dissolve very well into water. And so what you see is just a strong affinity for sediments that are at the bottom of a surface water body because of the organic content that are within those sediments. The most common legacy sediment contaminants that you'll see are polychlorinated biphenyls and polyaromatic hydrocarbons. Today I'll be talking a little bit about polyaromatic hydrocarbons, but certainly there is a full suite of different sediment bed contaminations. In general, the same characteristics that make these compounds have such a strong affinity for the sediment bed also makes them generally resistant to transformation. Depending upon where you are, and we've seen this across the United States at Superfund sites, contaminated sediments do tend to be contaminated for quite a while. They're generally resistant to biological transformation, other forms of transformation such as phototransformation, and because of that strong adsorption, they're typically not available in the water for other type of mediation processes. A couple of different categories that you can put them in. In general, all sediment contaminants tend to prefer the solid phase or tend to prefer the adsorbed phase to the aqueous phase, but the extent to which they prefer the adsorbed phase will dictate their ultimate fate. When solubility is extremely limited, they tend to accumulate in organisms, and these are the types of compounds that you see just bioaccumulating through the food chain. More problematic from a direct sediment issue would be the partially soluble contaminants, where in general they tend to adsorb to the organic fraction of the sediment beds, but over time will slowly leach out into the water column above. This is particularly problematic for sediment contaminants that are toxic at extremely low levels. So this one is one of my PhD student slides. She tends to really like pictures and she tends to draw just diagrams for everything. But I thought the cartoon worked in just kind of a general illustration. So for this presentation, we're mainly going to be talking about polycyclic aromatic hydrocarbons. So here's our model PAH spill. And we're looking at it from the sediment perspective, even though these are typically in solid phase, these would float because they're light. But you see it's strongly adsorbed to the sediment bed. And my picture on the right is showing a little bit of a close-up of that, where you've got these sediment beds and this pure phase illustrated in the picture in yellow a little bit before, and just in general, the dissolved molecules. In an ideal scenario, you will have microbial growth that's helping to oxidize and deplete these contaminants. But in reality, very few of them will actually oxidize and transform. And it's just due to really, really limited biodegradation. The thermodynamic considerations within sediment beds tend to be very, very low redox potential, which means that even though the majority of these contaminants will transform in the presence of oxygen, when oxygen is limited, transformation kinetics tend to be very, very, very slow. And you're more likely to see bleed out into the water column faster than you would see overall contaminant transformation. In contrast, we have a capped sediment bed. And capping is exactly what it sounds like. It would be putting a cap or a protective layer above the contaminated sediments. Here you can see in the diagram, this is a two-phase layer where it looks like there's sand on the very top and a gravel-like material towards the center. You can see my PAH is still contained within the sediment cap. And mainly what that sediment cap is doing is it's preventing the contaminant from leaching out into that blue water above. But additionally, it can provide extra residence time and a place for microbial growth, where with the increased growth and perhaps preferred conditions, you do see additional consumption of the pollutant and eventually cleaner water. And so really what we were looking at with this project is the extent to which capping material can influence the activity of native microbial communities. In other words, what could we put on top of these contaminated sediments that might improve the growth of selective organisms that would promote biodegradation processes? So just a little bit of background on capping materials. You can make a sediment cap out of a wide variety of materials, the most common of which is sand, just because sand is inexpensive and it's easy to deploy. You can also look at some slightly more expensive materials, such as carbonaceous materials, the most common of those being activated carbons. You could look at chemically modified clays, which I have in my diagram at the bottom left, where you can see it's a very different structure than your typical clay structure. It's just layers and these ionic layers that are designed to contain as many organic compounds as possible, as well as just general iron and iron filings. All of these have been demonstrated at field scale and all of which are fairly effective. In general, the main goal of this material placement is to delay advective transport. So delay the amount of time that it takes for these contaminants to break through the cap as they move upward through the sediments and into the waters above. Additionally, you can look at active caps, which will immobilize contaminants via adsorption. So in addition to just the fact that there's now additional material to travel through, there's additional sticky material to travel through. There's a number of different mechanisms that delay breakthrough of contaminants if you are looking at an active cap. Electrostatic binding is the most common. In general, to promote contaminant delay, you are looking at materials that are relatively small but have high surface areas, thus increasing the potential for adsorption, as well as the potential for organic matter to be present to help with that adsorption or sticking. Depending upon what material you're using, you can also use partially conductive materials. And I mention that because one of the most common materials being used to date would be activated carbon type materials, which are partially conductive. And most materials, particularly if you're looking at the carbons or the clays, you're looking at a highly concentrated surface functionality, which allows for a series of reactions to occur. From the biological perspective, the purpose of a biological cap would be to encourage microbial growth. And what you're doing is you're biostimulating. So increasing the presence or activity of microorganisms present. We know that most sediment systems naturally have organisms present that can degrade the contaminants, but the major issues tend to be access to contaminant as well as kinetic limitations. And so when you're looking at sediment caps from the potential of hosting favorable microorganisms, you're really looking at organisms that are already present, but perhaps aren't present at concentrations high enough to really make a difference. Depending upon your system, and I can think of several of these across the country, sometimes you can amend in vitamins, nutrients, preferred donors, preferred acceptors to promote that microbial growth. This tends to be a strategy that works better for groundwater aquifer systems where it's a little bit easier to deliver. If you're looking at a sediment cap system, solid phase is really a more desirable strategy just because you could immediately see release of anything soluble to the above waters and therefore not really affect your sediments themselves. Your sediments can be non-specific or can be dependent upon subsurface transport, which would reduce their efficiency and utility. Ideally, if you're adding something in, you want something that would be extremely targeted and extremely useful. But ultimately, and we see this across environmental microbiology, if you're looking to increase a particular organism that can degrade a compound of interest, the number one stimulator is typically that compound of interest, which in the case of a remediation scenario isn't really something that you're going to be adding just because you don't want to add any more of a contaminant if your goal is to eventually eliminate it. So looking at what has already been done with capping materials and what is generally known in the literature. First of all, if you're looking at lab scale, we have been able to demonstrate at lab scale, and several groups have done this, the increased potential of different microorganisms in systems that mimic a sediment capping system. Most of these studies have been done with chlorinated compounds, so you're looking at reductive pathways. There have been some studies that have been shown for the oxidative contaminant side, so the PAH side, with varying levels of success. What has been studied a little bit more in detail is this idea that placing capping materials in the field generally does affect your water quality characteristics. In particular, looking at the natural organic carbon content of pore water, that organic carbon content will change quite a bit when you add in sediment materials. What does that mean? Well, it means that you can have water quality information for the area immediately around a sediment cap. And when you go into the sediment cap, the conditions are going to be very different. Why does this matter? This matters because it affects the microorganisms that may or may not be present. And lastly, and I think my next slide shows this a little bit better, is that this idea of bioreactive capping technologies has been implemented at field scale. And so while this idea of a bioreactive cap is still generally in progress in terms of technology development, it is something that we've already observed partial success at field scale. So this is a slide that I was thinking of. It shows you a little bit better what's going on in terms of field. Most field scale bioactive caps aren't actually what I would define as a natural bioactive cap, because in the case of activated carbon, it is actually growing microorganisms of interest on the activated carbon system and delivering them directly. There's been a lot of studies shown that this is actually an effective way to get the right microbes into a system in a relatively short amount of time. However, the longevity of such a system and whether or not those microorganisms would be likely to persist over periods of time on the year scale is relatively underexplored. The reason I mention that is because if you're looking at bioactive cap systems or capping systems in general, typically your residence time for these contaminants is on the order of decades. And in a couple cases, you're looking at cap designs that are designed on the order of 100 years. So we are looking at relatively long periods of time. There have been a number of field scale studies that have pulled out cores from existing sediment caps, looking to see if there was microbial colonization, which there was. The question then becomes, well, are those microorganisms doing anything? And that's perhaps the most difficult question to ask because of the complex nature of a sediment cap. Because you're looking at forces such as adsorption and the different gradients that you see across the sediment cap, it's sometimes really difficult to determine whether or not anything is happening. These are extremely slow kinetics. These are half-lives that are can be on the order of years. And so when you're trying to differentiate very, very, very slow levels of bioactivity from background levels, it can be very difficult to discern, particularly if you have an absorptive material present. However, on the long scale of these capping systems, which, as I said, usually they're designed on the order of decades, if not a full century, looking at a half-life of 5 to 6 years is actually significant because it does suggest you will see attenuation of the pollutant over time. And so while we are seeing field scale information about sediment caps, we're not really seeing anything about the longevity of these capping systems. And furthermore, it's extremely difficult to discern whether or not these bioactive systems are actually effective, particularly when you're looking at a contaminant that's oxidative. Some of the reductive ones, those daughter products, are a little bit easier to track, and so there's been a little bit more success there. I mentioned already that the sediment cap environment is extremely diverse. When you're looking at these sediment caps, it's changing a large number of parameters, pH and ionic species, the bioavailability of hydrophobic compounds. You're kind of playing this cat and mouse game of you're trying to contain these contaminants, but you want them to be bioavailable. So to what extent do you want to contain, and to what extent do you want an aqueous solution when they're more likely to be bioavailable to different organisms? And there's conflicting accounts in the literature as to the effect of biofilm growth and EPS development over these different solids as to how it might affect contaminants that are absorbed on materials. So I'm going to go ahead and just summarize for you 3 of the PhD students' presentations or projects. When we set up her projects, we set them up at 3 different levels of caps. When you're looking at a sediment cap, the top part of the cap, and by top, I mean the part that is most exposed to the water column above, so the lake, the river, in some case the estuary or ocean, depending upon what system you're looking at. But that very, very top layer, and that's what I'm going to talk about in project 1, tends to be an oxic layer, and you tend to see high rates of oxygen delivery. As you go down into the cap, depending upon depth, you'll start seeing decreases in oxygen that typically occur 1 to 2 centimeters below the surface. You start seeing shifting biogeochemical controls, as well as towards the bottom where the conditions become highly reducing. And so that is the general schematic of the projects that I'm going to summarize here. The first one is looking at that very, very top layer, which is the oxic layer. The second project is looking at that intermediate zone where you are seeing shifting biogeochemical conditions. And the last one is largely anaerobic. So let's see if I can answer this. Are there any studies that show the most effective remediation of mixtures of multiple groups of structurally related but diverse organic toxins? So the answer is there are mixed studies on PCBs, and I don't know if this is quite the diversity that you're looking for, but just because there are so many different PCB congeners, there have been a number of studies that have looked at several PCB congeners at a time with mixed results. Typically in the presence of activated carbon, you tend to see higher rates of dechlorination with some of the higher congeners. So by higher congener, I mean more chlorine atoms. But once you get down to PCBs that contain 1, 2, or 3 congeners, there's less activity. There's a lot of work actively going on to optimize that process. To be perfectly honest, it's been about a year since I've dived into the PCB literature in that nature, so I'm not quite sure where they are, but that is ongoing. PCBs are the ones that I'm most familiar with. In terms of some of the other contaminants, I would say it's a little bit more mixed. It's been more of, let's see if we can get good results out of one of them rather than going full into the mixtures yet. So looking at oxic systems, and in this case, we were evaluating a number of different capping materials. We picked the 3 most common, looking at activated carbons, clays, and sands. Most of the data that I'm going to be showing in this portion had to do with the activated carbon and the clays, because both of which are absorptive. So, general experimental hypothesis is that we would see biodegradation promoted in systems that contain these absorptive materials. Our idea was that if the materials were absorptive, the pollutants would be very concentrated. The adsorptive materials would allow for prolific cellular growth, and we would eventually see a desorption gradient followed by transformation. We did examine both highly adsorbent capping materials, the highly adsorbent capping materials being the clay systems and the activated carbon systems, the low adsorption materials being sand and no capping. We used a couple of different models to assess whether or not this worked. Tracking, in this case, it was naphthalene, which is a model PAH decay. Looking at biomarkers associated with naphthalene transformation, looking at metagenomic data from the system, using 16S gene surveys, as well as just imaging growth. So here's our experimental matrix as to what we set up. And again, this is one of Giovanna's slides. And so one thing that she wanted to emphasize as a student who was in the lab setting all of these up is that this experiment, she actually had over 100 bottles because of the number of sacrifices she needed for the various analyses. But we are looking at systems that contain naphthalene, bioactive systems that are naphthalene free, and systems that are sterile, as well as the 4 treatments that I already mentioned. And then again, just different techniques for measuring. It was mainly biomarker and 16S gene surveys, as well as some imaging. And on the chemical side, it was looking at naphthalene decay and estimating rate constants. Wow, the image, okay, the image is showing up a little blurry on Zoom. It didn't show up this blurry in the publication. These are the results from the activated carbon in the clay systems. On the left column is tracking concentrations of naphthalene. My red lines are showing naphthalene degradation under bioactive conditions. My blue lines are showing naphthalene transformation under sterile conditions. And this was done over the course of 2 weeks. You can see in my activated carbon system, I'm looking at higher rates of, or higher standard deviations of my control, just due to the absorptive nature of the material. And in the clay, but in the bioactive system for activated carbon, you can see that within approximately a week, the naphthalene concentrations went down to 0. That's not what I observed in the clay system. The clay system became asymptotic after about the same period of time, and we didn't see any more changes within the 14-day window. And actually, she took this out a little bit further, and it just didn't change. We were seeing asymptotic behavior at approximately 50% transformation. My column on the right is showing biomarker. In the activated carbon system, which is the one on the top, you can see we're seeing an increase in the NaH biomarker, which is associated with PAH transformation inside the systems that contained the PAH. Inside the systems that didn't contain PAHs, which is my green, there's no biomarker increases. In contrast, you have the clay system, where we're seeing biomarker increases in both systems, regardless of presence of naphthalene. And from an environmental microbiologist perspective, this was really interesting to me because usually the contaminant itself will stimulate division and increase of gene copy numbers. So to see gene copy numbers increase in a catabolic gene that requires naphthalene in a system that didn't have naphthalene, which is my green system, was very interesting. And as we ran statistics on this, after the first 4 days, there was no difference statistically with that gene copy number in clay systems, either with the contaminant or without. And so we did see, in summary, we did see complete removal in the activated carbon systems, but about 50% removal in the clay systems. In the PAC systems, we were able to directly correlate naphthalene transformation with increase of the naphthalene degradation gene. But we're unable to do so inside the clay system. When you actually look at transformation rates, the activated carbon system was rapid transformation rates compared to the clay. What I'm not showing in this presentation in the interest of time are the other 2 systems that I talked about, the one with sand and the one without adsorptive materials. We did see transformation that went to completion in both of those, but at rates much, much slower than the degradation pathways that you see here. Microbial community that you see present. There's a number of different species that are shown in this diagram. What I'd really like to focus on is the difference between the purple, which is my magenta, and I don't know if my mouse is visible, but this magenta color that's in all of them and is particularly dominant in the activated carbon systems versus the yellow. And if you'll notice, the yellow is dominant in the clay systems and is present in the other 3 systems, but nowhere near the percentages that you see in the clay systems. In the clay systems, that yellow organism is dominating by approximately 75% if you were to average all the systems across. For all the systems, we've divided it into amended, which means contains naphthalene, and not amended, which means does not contain naphthalene. There's a lot of difference in the activated carbon systems, the media-free systems, and the sand systems between the 3 that contain naphthalene and the 3 that did not. That's not what we're seeing in the clay systems, and we did run a series of statistics, I think some of which are shown on the next slide, on the clay systems, and there was very little difference between systems, microbial community composition in systems that contain naphthalene and systems that didn't. The reason why I'm pointing out the yellow microorganism is because the yellow microorganism and the purple microorganism are from the same genus. They're both species of Pseudomonas. The difference being that purple species is a Pseudomonas species that is associated with naphthalene transformation. The yellow species is a Pseudomonas species that is not associated with naphthalene transformation. That's a generic Pseudomonas species. And so what we see in the clay system is the clay system increased concentrations of Pseudomonas very, very high. You can see from my figure, Pseudomonas concentrations are greater than 90% in almost all 6 of my clay treatments. But the Pseudomonas that is dominating is not the Pseudomonas that's associated with pollutant transformation. It's something very closely related, but just not an organism that's associated with overall transformation. We didn't see that in the other 3 systems. All 3 of my other systems were able to completely transform naphthalene over the experimental period evaluated. And the most prominent organism is Pseudomonas pedita, which is the naphthalene degrader. You can see it a little better on the different scaling options that I have. So this is a non-metric multi-dimensional scaling. In this case, it's Bray-Curtis. And so you can see here are my clay systems. Here are my activated carbon systems in red. And then here are my sand and my media-free systems. Statistically, there's very little difference between my sand and my media-free system. We saw the most variability according to the scaling in the activated carbon system. And you can see that the clay system actually clusters very separately. And there's not as much separation between the N's, which contain naphthalene, and the A's, which did not. Over on the left side, you can see there's different diversity indices of the different systems. In general, all the systems lost diversity, but you can clearly see a difference in diversity between systems that contain naphthalene and systems that didn't, for all systems except for the clay. We saw it in pictures as well. On my top here, I'm showing activated carbon systems. On the bottom is clay. And this is 4 days into the experiment. You can see that 4 days into the experiment, looking at my top left here, there is a full suite of organisms that are colonizing pore spaces inside the activated carbon. You don't see it in the control ones that didn't have naphthalene present, but you completely don't see it in either of the clays. This does change a little bit over time, where at day 6, we're looking at a much more robust community on the activated carbon system. And if you look carefully, you can see a couple of rod-shaped bacteria on the different clay systems, but there's no difference between systems that contain pollutant and systems that didn't. There's a few more organisms present in clay when you get all the way out to 11 days. At this point, however, the activated carbon is completely covered. And this is what it looks like overall, where you can just see the gradual increase in bacteria in the activated carbon systems that we just never saw in the clay. So in summary, we saw rapid naphthalene decay in the presence of activated carbon in sand, which is the data that I didn't show, as well as in a system that did not contain media. We saw high biomarker concentrations in activated carbon, and then normalized values were similar in sand and no capping. When I say normalized, it just means that overall the number of microorganisms was much higher in activated carbon, but the percentage that were able to transform naphthalene was approximately constant. And we saw quite a bit of attached growth in the activated carbon systems. None of these results were replicable in clay, which may be part of the reason why we just didn't see any contaminant transformation. Next study. So these are my shifting geochemistry conditions. And this particular study I'm going to go through pretty fast, just because this is overall a summary. What we were looking for in this particular study was whether or not these capping materials that we were examining would show any influence over microbial community composition and behavior within the first 100 days. Now, if you're familiar with PAH transformation under anaerobic conditions, it's extremely thermodynamically unfavorable and it's extremely slow. So we did not necessarily expect to see large amounts of PAH transformation in these systems because we were looking at anaerobic conditions. And while we did run a couple of tests, we really didn't see anything that was statistically significant. So I won't be reporting transformation kinetics here. But what we were looking at was, do we see anything that suggests this microbial community might change in a relatively short amount of time, meaning 100 days, which again, very short amount of time given the lifespan of some of these caps? Or is this something that really is going to take years or decades? So we hypothesized that we would start to see changes in that first 100 days, and that those changes would depend upon the type of capping material amended. We looked at 3 different electron accepting conditions: iron, sulfate, and nitrate. And again, we were mainly looking at 16S gene surveys to assess for success in this, just because the kinetic rates were so slow and we didn't think our analysis was going to be sensitive enough to see any transformation of the pollutant itself. Still looking at the same capping materials, we're looking at activated carbon, that organophilic clay, and then sand, and then the 3 different redox conditions. So, actually, I thought I had deleted this slide. I do apologize. All right, so just looking generally at the 3 different treatments, this is a nitrate system where you can see differences between clay, GAC, and sand at day 30 and day 100. I'm just showing one replicate from day 30 and day 100 on each of these, just because there were so many different gene surveys that were conducted in this one. Generally, what we saw was similarities between GAC and sand, and then clay had a very distinctive metagenomic profile, very similar to what we saw in the aerobic study. In all cases, we did see a significant shift from the baseline, and we did see it relatively quickly. The baseline being just native microbial communities that had been enriched for a prolonged period of time under nitrate-reducing conditions prior to this experiment being started. And then what we do see is a loss of diversity in clay systems that was very striking, where you typically see emergence of only 2 to 3 communities, and that was consistent across the board. Sulfate, we do see a little bit more diversity in the clay systems. Again, you're seeing general similarities between activated carbon and sand at the 30-day and 100-day mark, and a general shift from the baseline. Clay having a very, very distinct profile from the other 2, in this case, emergence of desulfovacta and desulfocapsa. Then lastly, looking at iron enrichments, where again, GAC and sand, and actually this one, when we ran statistics, we really didn't see much difference between the GAC and sand treatments, but a very, very distinctive clay profile. This is what they look like altogether. We did run a series of statistical analysis to look at what was more effective, the geochemical condition, so what type of electron acceptor was present, as well as the different type of amendment, meaning whether it was clay, GAC, or sand. We suspected that the geochemistry would play the most dominant role, and that's what you're seeing here. The higher those decimals, the more likely it is to play a dominant role. So those geochemistry numbers being quite high. What we were surprised at was exactly how high the amendment numbers are. So you are seeing significant influence of the amendment on the different types of microbial adaptations to those systems, but not as dominant as the geochemical ones. So while a high percentage of the variability was explained by geochemistry, you're also seeing a fair amount of the variability explained by the capping amendment. This is what it looks like on a multidimensional scaling unit, where similar to what we were seeing under the aerobic conditions, we're seeing a completely distinctive red circle around the clays. Clay would just behave very differently, and a fair amount of overlap with the different GAC and sand systems. And that was on both using Bright-Curtis and weighted unifrac distances, where GAC and sand are closely related, as well as we saw emergence in GAC and sand systems of organisms that you would typically associate with pollutant degradation. In clay, you tended to see very dominant organisms that we couldn't necessarily link to transformation processes. So in summary, we did see composition changing with media and biogeochemistry in a relatively short period of time, because that 100 days is relatively short. We did see that separate clustering of clays And while I didn't show my third figure here, we did see correlations with species in each of the electron accepting conditions present, particularly in systems that contain activated carbon and sand. So for our last experiment, we didn't do any more work with clay because all of our preliminary work suggested that the clay systems just really aren't likely to enrich microorganisms. And since our last experiment was looking at highly reducing conditions where pH transformation commonly takes years, given that none of our preliminary data suggested that we'd see any transformation, we did not include clay in these experiments. So we only looked at activated carbon and sand. And this experiment was conducted using radiolabeled materials. When you're looking at anaerobic transformation of pHs, the only way to track it is with radiolabeled materials, particularly if you're looking at a highly adsorptive material like activated carbon, which in this case was housing approximately 97% of the naphthalene. So being able to use that radiolabeled compound and to track CO2 in the headspace was really the only way that we could confirm for sure that we were looking at naphthalene-based transformation. So based on what we saw before, we assumed that activated carbon would impact biodegradation rates, consistent with what we saw on the first experiment, and that overlap that we saw between sand and activated carbon systems in our separate experiment was due to the relatively short experimental time periods, and that if we ran the experiment for much longer, in this case it was a 500-day experiment, we'd start to see separation of the microbial communities. We ran stable isotope microcosms. I'm not going to be showing that data. Those data were less conclusive than the radiolabeled naphthalene microcosms, which are the data that I will show. And we did look at both total naphthalene, 16S gene surveys, and then mineralization data. Again, simple microcosms that were representing capping systems. In this case, only 3 treatments, the activated carbon, the media-free, and the sand. As well as biologically active, sterile, and systems that contain naphthalene, or systems that did not contain naphthalene. So fate of naphthalene that we see in these systems, first of all, over the 500-day experiment, we did see tailoring off of naphthalene concentrations in both the sand and media-free systems. However, in the activated carbon systems, we do see clear separation between the systems that were biologically active and the systems that were sterilized using azide. When we looked at basic statistics, we did not see a significant difference between the sand system and the media-free system in terms of naphthalene transformation. However, the difference between the sterile and the bioactive activated carbon system was statistically significant. Looking at headspace. So when we're looking at headspace, we're looking at radiolabeled carbon dioxide. In these systems, the only source of the radiolabeled carbon dioxide would have to be the radiolabeled naphthalene. So if we're looking at radiolabeled CO2, it means that the naphthalene was fully oxidized to carbon dioxide. We only saw radiolabeled carbon dioxide in one system, which was the activated carbon system, consistent with our bulk naphthalene data. So we did see a significant accumulation of radiolabeled CO2 in the headspace of activated carbon compared to sand and media-free, which spiked at certain time periods, but were many orders of magnitude lower than what we saw in the activated carbon system. A significant amount of work went into modeling this, and it's because it's a dynamic system. You have a system that is adsorptive, biotransformative, and by looking at some of the non-adsorptive system, you can tell that there's abiotic reactions, possibly hydrolysis going on in the system. And so this was the part of the work that I mentioned at the beginning that was a significant amount of mathematical modeling and different transformations. I'm not really going to go into the details here, but I will say that overall the system was divided into 3 different phases using the activated carbon systems. The first 162 days being a lag phase where we really couldn't tell any difference statistically in the naphthalene data. The second portion being an exponential decay period where we are seeing first order transformation rates of naphthalene in systems containing the microorganisms. And then lastly, an asymptotic phase where the main contribution, and actually you can see a little bit of increase in naphthalene in the aqueous solution, which we believe was due to slow desorption rates finally catching up. Shifts in microbial communities. Again, this time, and these data were taken at the final 500-day mark. At this point, we can see a clear separation, not as clear as some of my other figures, but the activated carbon systems are starting to shift out from the overlapping media-free and sand systems. We didn't see a whole lot of differences in diversity between media-free and sand systems. You do see a little bit of variation in those diversity at the 3 different time points that we analyzed. Overall, the activated carbon systems were less diverse compared to the baseline enrichment and were less diverse than the 2 non-absorptive treatments. But the main take home was that you did see slow separation of microbial communities over time, which did go back and prove our initial hypothesis, which was that we would see a separation between those activated carbon and those sand systems if we allowed it for a timescale that was more appropriate for an anaerobic environment. Ran a couple of different correlation factors, which I'll go through really quick because we're almost out of time, which is when we saw increases in Geosulfa virga and Geobacter, we saw increases in headspace radioactivity, as well as increases in sulfide, suggesting that when these microorganisms were present, there was more sulfate reduction, which is the electron acceptor, and more 14CO2 produced, suggesting a naphthalene was used as a carbon source. 2 other organisms that we saw statistically significant increases in, Acidomycobacter and Acetobacterium. We saw increases in these organisms when headspace radioactivity increased, but they were not related to sulfide or sulfate reduction at all, suggesting that these organisms, which are within Clostridia, were enriched centrophically, and looking at the literature that is similar to what other groups have reported in regular groundwater systems, so non-capping systems. So in summary, what we saw over the 500-day experience is that as aqueous naphthalene decreases, we saw increases in gaseous radioactivity. We saw enrichment of sulfate-reducing bacteria that we could correlate to metabolite production, and were consistent with the literature suggestion as to sulfate reducers that are capable of using PAHs as a carbon source. And overall, we saw these most pronounced in the activated carbon systems. The other 2 systems were not statistically significant, but the system that showed all of these consistent was the activated carbon system that showed considerable mineralization. So what does this mean for design? Well, it means that if you're looking at a design, you really have different considerations to take at any point in the layer. My first diagram up here with oxygen going to CO2, that was the first project where you're looking at just the surface and transformation. And activated carbon promoted that the most out of the 4 treatments studied. You do see some transformation in that middle zone where you're looking at a series of different electron acceptors, but influences of the different capping materials with the idea of promoting good microorganisms that are capable of biodegradation occurred most prevalently in the clay and sands, in the activated carbon in the sand systems, and not the clay systems. But certainly when you get to the very bottom of a cap where transformation is very unlikely to occur, we were able to observe transformation over the course of 500 days at significant levels, but only when activated carbon was present as a stimulant. So this is a very quick summary of what I just described in that in different words. But I'll go one step further and to say that this is not comprehensive in general, because we were looking specifically at controlled environments that had very little organic carbon present, and our systems contain sufficient nutrients. Really, when you're looking at this in the field, there's a number of other considerations that would very much need to be taken into consideration. The first is whether or not capping material placement is affecting nutrients. And field scale has observed this. If you're looking at low nitrogen, low phosphorus conditions, because activated carbon can absorb both of those, you can actually pull out necessary nutrients and prevent biological growth, but this largely hasn't been explored. We didn't talk very much in this presentation about kinetic limitations, absorption, desorption. That is certainly coming into play. We saw a little bit of evidence with the longer term experiments of just the extent to which slow desorption rates off of highly adsorptive materials can curb your biological reactions. That's something that has been continuously mentioned in the literature, but overall is not well understood. And then lastly, this idea of syntrophy and cells working together, as well as organized biofilm networks and formation that formulate uncapping materials. A little bit of work has been done on this from the PCB side, but not very much in the terms of oxidative contaminants like the ones that I talked about today. So overall, thank you very much for bearing with me. I'm just looking at my watch. It looks like I was just about at 40 minutes once I got started. So I hope I haven't kept you all too long. This right here is my lab group, and we're not actually that big. That's a whole lot of spouses as I'm looking at the table there. This was one of my students' birthdays. And of course, well before COVID, because there's 12 of us at a table and we're all eating Indian food. But we go out a couple times a semester, or we did prior to March of 2020. And I hope that soon we'll be able to continue that, as well as Giovanna specifically, who's in the back of this photo, but there's a blown up photo of her here, the PhD student who did all of this work. Particular call out to Kayleigh and Aldrich, which is a consulting firm located out of Connecticut. One of the engineers from Kayleigh and Aldrich obtained a lot of the materials for the study, as well as just provided a very practical engineer's perspective on, what would you really need to know if you're a field scale engineer and a diver who's putting in these types of systems. And then the majority of the funding for this work came from the Electric Power Research Institute. Oh no, my picture left. So usually there's a picture on here of my crying baby. That was the picture of the baby that I promised earlier, but thank you very much for your attention, and I'd be happy to answer any questions that you have.
Well, this is our thunderous applause for you, Kay, and Zoom meeting. You can't hear the applause, but it's going on virtually. So any questions for Kay from the audience?
All right, so on a scale of 1 to 10, guys, how fast did I talk? I tend to talk very quickly, and I don't necessarily notice it all the time.
I have a question, Kay. Eric Segrin here. Thanks for your seminar. You alluded to this a little bit at the end, but obviously the relative rates of various processes of interest here with desorption, absorption, and diffusion and biokinetics are important. And is that part of the work that you didn't report on today, or is that something you guys did not really dig into?
So the last work, the sulfate-reducing work, we had sufficient work, data to calculate each of those rates, specifically the desorption rate. We were looking at it from the perspective of orders of magnitude. Are we the same order of magnitude? Are we a different order of magnitude? Those values are in that publication. To be perfectly honest, I don't have my list of rates here. What we saw was that based on the information that we had, the desorption rate was not limiting until we got to the lower concentrations of naphthalene. When we saw just limited transformation, when we saw aqueous concentrations, because the bulk of it, about 97% in that study was in the sorbed phase. Once about 50 to 60% of the aqueous phase depleted, that replenishment was taking quite a bit of time. And so that one was approximately the same order of magnitude as the degradation rate. So as it dropped, that desorption became a lot more significant. The abiotic rates that we saw, the abiotic transformation, just the losses that we saw across the system over the 500 days, those were approximately 10 to 20% of the biotransformation rates. So not small enough that they're insignificant, but small enough that it was easy to discern the difference between biotransformation and just natural processes such as hydrolysis, and possibly had space losses, even though we thought the bottles were sealed pretty well. Let's see, other rates, and I'm trying to think off the top of my head. But no, we did actually go through quite a bit of discussion about rates. And in particular, getting that paper published, we had a reviewer who had a large number of questions about rates. And so we went back and forth, trying to make sure that we were able to make the best possible estimations given our data and the complexity of the system.
Okay, thank you.
Any other questions, maybe from the students?
Well, I'd like to follow up on Yarik's question, if I could. Kay, I appreciated the talk very much. I enjoyed that. This is Noel Urban. So following up on Eric's, what about the rates relative to diffusion? I mean, you postulate reactions going on at different places, the surface of the cap, the middle of the cap, and the bottom of the cap, but that depends on there being transport through the cap. And I'm just wondering, how did your rates compare to diffusive transport?
To be perfectly honest, we did not examine diffusion. In terms of advection in an actual capping system, you could look at a scenario where advection is really going to drive, or scenarios where that upwelling, that upwards movement of water is so slow that you don't necessarily see strong advective forces, in which case your diffusion is going to come into play quite a bit more. I didn't mention this in the description. In the aerobic systems that we studied, the 14-day experiments, they were continuously agitated to abate the possible effects of diffusion, to minimize whether or not it was actually going to be diffusive limitations that prevented. We knew that there was nothing we could do about desorption limitations, kinetic limitations, but we tried to minimize diffusion. We did run a series of bottles on the shaker and without the shaker, but with sufficient oxygen. So those ones we had to continuously replenish oxygen in to make sure that the system didn't drive itself anaerobic. And we didn't see a lot of difference in transformation rates in the systems that were continuously agitated and the situations that were stagnant, suggesting that at least at the scale that we were looking at, which was small scale lab, we weren't seeing significant influences of diffusion. And so we did not consider diffusion in the 480-day experiment just because it was a longer experiment. And because we didn't see a lot of influences at the smaller experiment, we assumed that because the larger experiment was consistent, that we wouldn't see a lot of limitations. But absolutely field scale, if you're looking at very, very slow advection, diffusion is going to very much play a part, as well as different parameters like a preferential flow path. Of course, you're going to have issues with depending upon what type of cap it is and how well it's compacted. I don't know if that fully answered your question, but I would say it was considered, but not necessarily investigated at length.
Thank you.
Any other questions or comments for Kay? I had a question, Kay, and that is, so the original bed level before you put a cap, then you put the cap on and there's some treatment going on. How far below the original bed does the treatment be effective? Am I explaining it right?
So if you're putting the cap on top of the contaminated sediments, because water is usually going up, so you're laying it between the sediment and the water above. It depends on the type of cap and the type of material. Most sand caps that I've seen have been on the order of 6 inches. I'm aware of one cap where it was on the order of 3 feet, so it can be quite a bit deeper. In the case of activated carbon, that's usually a polishing step. So just a little bit on the top, in which case you start seeing mixtures of sand and activated carbon. We did a little bit of exploration into that to see whether or not those mixtures really had a significant effect. When I ran the statistics on it, we really weren't seeing too much of an influence. What it did suggest was that to really better investigate it, it would need to be very systematically looked at, and it was a little bit beyond the scope of the project. But you are looking at, in the case of activated carbon, pretty thin layers because of the cost. Sand, as I said, 6 inches seems to be the norm, but I have seen, closer to 1 meter. And that one's pretty easy to deploy. It's just blowing sand on from a boat above. With organophilic clay, those tend to be pretty thin and they tend to be compacted. One of the issues with organophilic clay is making sure that you've compacted it correctly and you're not creating cracks that would therefore allow just like rupture of your system. So I don't know, did that answer your question?
Yeah, I think so. Oh, I see some in the chat here too. Can you see the chat, Kay?
All I see is Audra's comment of thank you, Kay.
Oh, yeah, great. Any last questions before we go? We're past 4 o'clock here.
You guys, I do apologize for running over. I'm sure you guys are tired and hungry, but I appreciate you listening to my talk. And as I said, if any of you have an interest in presenting in the spring, and I may email Brian directly on this, certainly TTU would be very happy to host you. Our seminar, unfortunately, is also going to be on Zoom, but you know, we certainly be interested in hearing topics that are perhaps outside of our university and our regional area of the country.
All right. Well, thanks everybody for coming, and thank you, Kay, again for taking the time to enlighten us. So thanks very much, everybody.
All right. Have a great day.
Too.